Hi, Mrs. Murdock, and this is a review for the aerobic respiration part of the test that you're going to be taking on Wednesday. So aerobic respiration is what I'm going to concentrate on right now. So this diagram is a flowchart that I gave you that I had you fill out on your own, and I put the key over there, and you may have filled it out, but I want to make sure that you really understand it well, okay? So let's concentrate on the breakdown of glucose first, because we know probably we're more familiar with that one. So the first thing that happens to glucose in here is glycolysis. No more mysterious than that, right? To break sugar, glycolysis, right? And this process number 8, takes the glucose and breaks it down into 2 pyruvates, which is all number 12 is, right? This is the pyruvates, or 2 pyruvic acids. And remember, glucose has 6 carbons, and each of the pyruvates has 3, right? So if we're doing that. And you can get 2 ATP out of this process. I don't know what that is really doing there. I would really rather put NADH in there because I think it's more useful. Okay. And you know that this is a multi-step process which has an investment phase and a payoff phase, which, you know, it gives you a net of 2 ATP, but you may remember the reason why you call it an investment phase up here is because you're using up 2 ATP here before you're getting 4 down here, right? And that gives you that net of 2 ATP, which is very useful, and if you're just a little bacteria or a tiny little cell like that, that might be enough for you, right? And if you didn't have oxygen, then after glycolysis, the pyruvates would enter some form of fermentation. But in this case, we're going to go on and pretend that we have oxygen and we have a mitochondria. So the next thing that happens after that is those pyruvates are further oxidized in the mitochondria, right? So down here, now we're down to the mitochondria, and they become acetyl groups, 2 of them, which connect to a coenzyme A, right? So each one of these is 2 carbons, not the coenzyme, but the acetyl group. And there's a little bit more of oxidation going on there. You do get your first carbon dioxide from the process, from this, which is really the link reaction. This is the link reaction here. And everything from here on out is inside the mitochondria. Okay. All right. And if you keep going, oh, and the other thing I will say is that you also get, if you remember, you also get 2 more NADHs here. Okay. Keep going. So number 10 is the Krebs cycle, also known as the citric acid cycle. They're both the same thing, so don't let that confuse you. The Krebs cycle takes that acetyl coenzyme, those 2 little carbons, and further oxidizes them down. And again, you get some carbon dioxide from that, which are just wastes, not too important. You get 2 ATP from this, just from the Krebs cycle. Remember, we got 2 ATP from glycolysis and 2 ATP from the Krebs cycle. Both of those events right here and right here at this point, well, actually in the payoff phase, sorry, in the payoff phase. There's only certain places in these these long series of metabolic steps, right, where something called substrate level phosphorylation happens, right? Substrate level phosphorylation. And if you've really been reading the book, you know what I mean. So substrate level phosphorylation is not as efficient, not nearly as efficient a way of making ATP or phosphorylating an ADP into an ATP as what happens in the electron transport chain. Electron transport chain is called oxidative phosphorylation. We talked all about that today, right? Substrate level phosphorylation is when you have an enzyme at this point and this point. There's only like 2 or 3 points in the whole metabolic steps where there happens to be an enzyme and there happens to be a substrate with a phosphate sticking on it. And the enzyme fits with that substrate. And it takes the phosphate off the thing and puts it onto ADP. Hence, substrate level phosphorylation. Okay? So that's that type of phosphorylation. You don't get very much from that. But that does happen during the Krebs cycle. Okay, the other thing that happens, the most important thing really though that happens during the Krebs cycle is you get these things. These things are very, very lucrative, right? The NADHs and the FADHs are electron carriers with high energy electrons that came from food molecules. Ooh, there. Okay. So all these organic food molecules from the glucose on down, every time it was broken and broken and broken and broken, the electrons that came from those broken bonds got caught by these guys, NADHs and FADH2s. So now they're loaded with these high energy electrons. They're going to go into the electron transport chain, and they are going to make the most ATP. Got red just like I did before, right? And this is not substrate level phosphorylation. This is called oxidative phosphorylation. My face out of the way. Phosphorylation. Whoop. Okay. And that's where you're in the mitochondria and you have that intermembrane space and you're pumping it full of hydrogen ions. And then the hydrogen ions are coming through and making lots and lots of ATP through all those ATP synthases that are waiting for them to come through them so they can phosphorylate the waiting ADPs in the matrix. That's called oxidative phosphorylation because you're using electrons that were oxidized from food molecules to power that phosphorylation step. And you can get much more ATP out of that than you get from the substrate level phosphorylation that came with glycolysis, which was the pathway that evolved first, right? Evolutionarily, it evolved to make those enzymes in the cytoplasms of all living cells. They're still with us now. Those same exact enzymes are in our cells that were in those bacteria cells 2 billion years ago. Glycolysis is very old. It's very conserved. It was just a question of once you got to those pyruvates, then what do you do? Do you just ferment them so you can keep those 2 ATPs going? Or do you figure out how to handle oxygen in an efficient way that gives you a way of phosphorylating a lot more ATP per glucose, right? So one glucose molecule using this full thing with the mitochondria in place and oxygen as a final electron acceptor can give you 36 to 38 ATP per glucose. But without the mitochondria, without the use of the oxygen, you can only get 2 ATP per glucose. Right? And again, if you're a tiny little cell, that's fine. But if you're a big, expensive organism like a tree or a human, you're going to need more. Okay, so that I think is where I'm going to stop recording this because I'm going to go on to some other things here in a second. Well, actually, maybe I'll add a couple more things here. Okay. So now let's go to the top and just look at proteins and fats. Let's go back up here and now think of it from the perspective of, well, what if we're looking at protein and fat? How does that work out differently? We have just enough time for that. Okay. So let's look at this. Let's look at fats first, right? Way back in the biochemistry unit, you know that a triglyceride breaks into two things. It breaks into a glycerol, and it breaks into fatty acids. Fatty acids. A glycerol is a 3-carbon molecule with 3 OH groups on it. And that lends itself very nicely to be turned into, I think it's just turned into PGAL, which is also a 3-carbon intermediate that's in glycolysis. So it's transformed into this and then it carries on through glycolysis, right? Fatty acids, we're talking about lots of carbon and hydrogens, right? Entire chains of carbon, hydrogens. It's a hydrocarbon, right? All like that with hydrogens at every free spot. So what the cell does with these is it cuts them into 2 carbon units. 2 carbon, 2 carbon, 2 carbon. So with 2 carbon units, where do they come in? You can take a 2-carbon molecule and turn it into an acetyl group, right? Which has 2 carbons, kind of like, acetic acid is just vinegar, guys. That's all it is. So an acetyl group is 2 carbons with hydrogens on them, and you can convert that easily into an acetyl group, which connects to a coenzyme A, and then you skip glycolysis entirely and go straight to the Krebs cycle. Okay. So that's what happens to the fat molecules. If we go back up to the protein molecules, hopefully you'll remember this too from the first unit. Remember when I talked about when proteins are digested into their monomers, those monomers are called amino acids. Remember that? Amino acids is number 9, right? And you've got digestion here. Digestion of proteins, digestion of fats. If you cut them into their component parts, this is what you get. So with the amino acids, remember, amino acids can differ, right, from amino acid. There's 20 different kinds of them. And they all have different R groups. And if the R group has 3 carbons on it, a 3-carbon R group could be converted into a pyruvate, which also has 3 carbons. If that, if the R group has, get this out of here. If the R group has 2 carbons on it, there's 2 carbons R groups sometimes. If it has 2 carbons, it'll be converted to an acetyl group, just like the 2-carbon fatty acid groups would be. If it has 4 carbons, if you really were brave and went into the book and looked in detail at the Krebs cycle, a lot of the last 2 intermediates here and here are 4-carbon organic molecules. So those can enter the Krebs cycle there and help it keep going, right? And then remember deamination? Deamination, which is you take, remember this? There's the R group. Remember the NH group? That is ammonia, right? Deamination means taking off the amine group. That's exactly what it means. And we actually talked about this when I talked about human excretion. And where does that go? Into your urine, right? And you pee it out. Just like carbon dioxide goes into your lungs and you breathe it out. Whereas that you need. The most important part of the whole thing. Okay, that is as far as I'm going to record here today. I hope that was helpful.